You're welcome back to the second session. Today we'll be discussing about the genetic code and the gene structure. And here we'll be talking more of in the aspect of molecular biology. Here we'll be discussing about how amino acid sequence arise from nucleotide sequences, and also what we mean by the promoter sequences, introns and exons, and the reading frames. Now, there are some key concepts we need to understand from our previous class. We have the DNA transcribed into RNA and RNA transcribed into translated into protein. That is the way in which the information is passed from the DNA to the protein, which is the functional part, which is the central dogma. Now, here, the cell stores the information, as we all know. The transcription and the translation process occurs, and then the nucleotide of our messenger RNA assembled to the DNA, and all those processes, we all know them. So now, during the process of replication or transcription or translation, mutations in DNA are passed. That is, mutation can be a form of mistake in the copying process. It is passed from the DNA to the RNA and then to the protein, which results in variation and then are acted upon by natural selection. So these are some of the stages that we have to understand. So we can read that up also. Now, for a DNA to be read successfully, you all know the process. Now we now all know that the process that is involved is also complex, not simple. So these are also important for us to note that the DNA is a simple language composed of 4 deoxyribonucleotides known as the A, C, and T and G's. It is different in the case of RNA. We have A, C, U, and G. But based on what we want to discuss today, how the nucleotide, amino acid are gotten from the DNA sequences. Now, DNA information is packed into units called genes. We initially made a definition of genomics, genetics, and the likes. So what is a gene? A gene is a linear sequence of deoxyribonucleotides that are necessary for converting a portion of that sequence into a complementary sequence of ribonucleotides inside a cell. That is, a linear sequence of DNA that are necessary to convert into a sequence that are complementary to RNA. Now, we would understand better. So there are always a single linear sequence of deoxyribonucleotide on a single piece of DNA, and the average length of a gene is 10,000 to 15,000 base pair. 10,000 to 15,000 base pairs. Now, genes are transcribed into RNA in a process called transcription. We know that already. And a portion of that gene that is replicated in RNA is called the coding sequence, while the ones that are not translated are non-coding sequence. It is very simple. Now, in the structure that we have here, in the figure we have here, so the smallest functioning unit of a DNA is a gene. So here we have different genes on the line of a DNA, different genes. So, and in a gene, you can have an intron or an exon. You can have an intron or an exon. Now, and at the edge of this intron or exon, you have what we call the regulatory sequence. So, which is always at the beginning and at the end of a particular gene. So let us assume this is a mitochondrial gene. The mitochondrial gene. You can see how long it is. Let us assume. So now you have this particular gene in the mitochondrial genome. So let's say this is the CO1, this is CO2, this is ND5, and the likes. So now you have this particular gene. Here you have the exon and the intron. So what do we mean by intron and what do we mean by exon? Now, as you can see in the final replication of the RNA, which is the messenger RNA, the final replication, you can see that these introns are missing. They are missing from the final replication. So introns are like gaps between the exons. Introns are like gaps between the exons. So whereby in the final product, all you'll be having is the messenger RNA. In the messenger RNA is the exons and also the promoter sequences and regulatory sequence that are at the beginning and at the end. So that is simply how this process is carried out. Now, the coding sequence is often broken into segments called exons and are separated by non-coding sequence called introns. So that gives us an overview of what we are talking about. So we explained earlier also the different types of RNA that we have. We have the ribonucleotide RNA. We have the messenger RNA, ribose RNA. We have the transfer RNA. We also have all this different RNA. So, but this is beyond the scope of what we want to discuss in this session. Now we have the genetic code. Now, what do we mean by this genetic code? Now, they have key characteristics of the genetic code. So the first one is that each triplet codon is recognized by a transfer RNA, which is associated with a cognate amino acid. That is, when you have a DNA bases like the A, G, C, T's and the likes, and they are translated into they are transcribed into RNA, and you have these different sequences. Now, 3 of those will make one codon, 3 of those will make another codon, and that is, they are all in triplets. They are all triplets. A codon is made up of 3 nucleotide bases or RNA base, nucleotide bases. Yes. So, as you can see here in the picture. Now, out of 64 potential codons, 61 are used to specify the 20 amino acid building blocks of protein, whereas 3 are used to provide chain terminating signals. Now, for students of biology, we all know that there are 20 amino acids in living cells. So, these 20 amino acids are formulated with this particular formula, which is the genetic code. Now, as you have this first codon GCU, it has a name. ACG has a name of the respective amino acid that is decoding. So, because there are 20 different amino acids but 64 possible codons, so most amino acids are indicated by more than one codon. So we have gotten the explanation surrounding that. Now let us go back to the explanation. Now, the genetic code is a set of rules used by living cells to translate information encoded within genetic material. So it's a set of rules, basically, just like we've explained, and the translation is accomplished by the ribosome, which links the proteinogenic amino acid in an order specified by the messenger RNA using transfer RNA. Now we are here telling us the explanation of the different function of the RNAs. So the transfer RNAs are used to carry amino acids and to read the messenger RNA, 3 nucleotides at a time. So just like their name, transfer RNA and messenger RNA, the name denotes their function. Now, the genetic code is highly similar, but there are variations within different class. We have different genetic codes. For example, the genetic code for vertebrates is different to that of invertebrates. The genetic code of invertebrates is different to that of a ciliate. We have that of yeast. We have like 15 different genetic code. So which means that, let's say we have a GCU coding for alanine in vertebrate. It might not be GCU that will be coding for alanine in invertebrate. So that is the simple explanation for that. Now we have the universal genetic code and we have the vertebrate genetic code, and we can see the differences between the two that are indicated in the red colored. So in the case of universal, AUG always signify the start codon, which is the methionine, whereby here we have two start codon in the vertebrate, which is the AUA and the AUG, and the stop codon is denoted by this two and this two, and then we have tryptophan that are different to the one here. This shows another example of the different invertebrate genetic code that we have. Now, this difference in genetic code will lead us to what we call mutation. So these mutations have their uses. Mutation has the definition. So a mutation is an heritable change in the DNA sequence of an organism. That is, it is transferable from the parent to the offspring. So the resulting organism is called a mutant. And as you can see in this figure, there are different types of mutation. We have the point mutation, which are indicated by the silent, missense, nonsense. And we also have the insertion or deletion, as you can see in the figure here also. Now, what is, okay, like we've said, let's go back to the explanation here. It says one type called point mutation affects a single base, and it mostly occurs when one base is substituted or replaced by another. So, and whereby we have another one called insertion, whereby one or more bases are inserted into the gene, or one or more bases are removed from the genes, and that is called one or more bases are removed from the codon, and that is called a deletion mutation. So that means we have like three types: point mutation, insertion, and deletion. Now, in the first figure here, we have the silent mutation. Now, a point mutation may be silent, and it can be a missense, and it can be a nonsense. So when it is silent, when a mutation is silent, you can see, just like we explained earlier, that there are different codon might code for a particular amino acid. A different codon might code for the same amino acid. Now, in this case, we have valine. We also have valine here. So the DNA is AGCGTA, while in this case the GTA is translated as GUA. Now we now know that anytime you see a T in the DNA, it will be a U in the RNA, because it is switched to uracil in RNA. So GUA is a valine, while GUU is also a valine. There are two different set of codons, but they are coding from the same protein because of the genetic code that we have. So here, this GTT, this is GTT, this A is switched into T, but still we'll be having this particular result of valine. That is a silent mutation. No effect, nothing. When you have a missense, it will give you a totally different amino acid. So here you have proline, CCC, CCC. Proline here, ACC will give us ACC, which is threonine. So this is another type of amino acid. A missense gives you a different amino acid, whereby a nonsense leads to a stop codon being initiated. That is, it breaks your sequence into bits. Like instead of the gene to have a full gene, like let's say the gene should be 500 base pair long, and you have a nonsense in your sequence or in your translation, the gene will be shortened. Maybe the 500 will be broken down into 3 parts, which will not make it the gene any longer. Now we have the case of insertion and deletion. So here we have serine, valine, proline, threonine. So when this TA is removed, it will shift the codon up forward, and then having different interpretation, so which is also necessary for us to understand. Now, point mutation leads to different types of effects, and one of such effects is the manifestation of a disease. And as we have the sickle cell anemia is a popular type of point mutation that leads to a particular disease occurrence, which is the sickle cell disease. So we have the GAG, which is the normal hemoglobin, and then it is when there is a single point mutation, we have GOG, which is leading to valine. That single point mutation leads to this disease effects. There are other effects of point mutation that we might not be able to take in this particular session. You can read forward on that. Now, questions might come up regarding the mutation rate in human genome. And this mutation rate in human genome, it occurs once in every 10,000 to 100,000 base pairs. We know that we have 3 billion base pairs. So now we know when we say this mutation occurs between once in the 10,000 to 100,000 base pairs, it might look infinitesimal, which might look little, but the effect might be huge, as we've seen in the case of sickle cell anemia. Now let us move on to reading frame. So we've known how to read frames from our knowledge of codons, but how do we read frames in a particular set of genes, a sequence of a gene? Now, as you can see here, we have the sequence of ATP8 and ATP6 following each other. Now, when you take the first 3 letters, as we know that a codon is a particular codon is denoted by 3 nucleotide sequences. So we have AAA. Let's say this is the first one, second one, third one. It will give us the K. But when we continue like this, we would not have the start codon to initiate the translation of the particular gene, which is not good for us as for before any gene is the translation of that gene or transcription of that gene begins, it will have to start from the start codon, which is the methionine. So when you start from the first sequence, you would not get very good translation. When you start from the second one, you will not have a good translation. But when you start from the third one, you can see ATG straight up. That is the start codon. It will give you the interpretation of, it will begin the translation of that particular sequence. So we have it here. So once we have this M, then that means we are good to go. And up to, we'll get to a stop codon. So that is when you begin. Now, okay, that is for ATP8. So that is how to read a set of genes. So that is how to read a set of genes, and that is called the open reading frame. The open reading frame is the point at which the gene is, you begin reading the genes. So it is defined by the initial triplet of nucleotides from which translation starts. It sets the frame for a run of successive non-overlapping codons, which is known as the open reading frame. So that is a short explanation about that. Now I put out an explanation, an example of different open reading frames, the allocation on the genome, the spaces between different genes, in the case using Anopheles sinensis as an example. So Anopheles sinensis genome is 15,422 base pairs long, especially the one like 15,000, approximately 15,000 base pair long. So there are different lengths depending on the type of individual you sequenced, or from the location you sequenced it from. So in this case, we have a transfer RNA starting from the position number 1 to 68, transfer RNA glutamic acid starting from position number 66 to 34. You can see that if you do not have an idea of what the open reading frame is, you might not understand the whole thing behind this arrangement. Now, the first gene, which is the ND2, starts from 203 and stops at 1228. So as you can see, this is another gene, this is another gene, and here we have the start codon of every one of them, and then the stop codon of every one of them. So we also have the number of intergenic nucleotides, that is the spaces between each and every one of them. So this is the full information on the NCBI website, and then we know what start codon and stop codon is at this time. And one thing that we might not have had an idea of what it is the promoter sequences. So these promoter sequences define where transcription of a gene by RNA polymerase begins. So they are very, very important. So as you can see in this particular figure, you can see that transcription start site, then immediately you see the start codon. The promoter sequences immediately follows the start codon, the stop codon, after the coding sequence have been written. So promoter sequences are very important. Now we know what introns are and exons are, so from our previous class, so we are good to go.